Good morning, everybody. Today we're going to continue a little bit about diversity functions. Last time you were calculating by hand from scratch you were exploring the data set, this Estes data set. And today I am going to start with a short intro to what we mean by functional and phylogenetic diversity. But first, I don't know if you hear me well. Hello. Hello. Huh? I can't hear myself, so it's horrible.
We can hear you.
Okay, thank you.
What are those slides?
They're not there. No, yeah, the site is not updated. I just got my 4th dose of vaccine, and I had 24 hours of fever in the last days, but viva Osus, and I was just like very sick this weekend. But I will update this. I will update this with better examples than today, and I'll let you know. So basically, so far, you should have your repositories should be organized. I had told you about a map made in TMAP for the spatial analysis class. And we had some code with calculations from last class that was mostly in the notepad, but I also expect this to be in the repo. During the week, Sarah and I, we will be checking the repos, because we are talking about evaluation for you all. And in this case, in this class, of course, what we want you to know how to do is creating repos, updating them, organizing your work. The workflow part is key for this, more than knowing exactly how to do some calculations in R, or what we want you is to have the good practices in how to work. So this is key. It's a little bit different from maybe other modules that you're having, but this is exactly what we want. What we want you to do is to organize yourselves and to be using Git and to be using your computer in clean, organized ways that should be useful for yourselves in your other work. If you have any questions, I will be here anyway. I was just checking the code for you, the ones that you put in the notepad. And I saw many creative solutions for the exercises last time. And the next step would be to create functions out of it. So if you create a function that will calculate Shannon diversity, this is not needed actually, because R has ways to calculate Shannon and Simpson diversity. I will just take this off. Are we okay? Don't we have like any symptoms? Anyone has any? It's because I can't hear myself. It's horrible. Oh, hi. Thanks for wearing the mask so I can do this. I was checking some very creative functions and very creative solutions that you had. I couldn't have the time to check if they were correct, but most of you had different ways of solving. They're in the Etherpad. And when we think about creating functions, which would be the next step, in this case, we don't need a Shannon diversity function because R has. But I would like you to think about the application in real life of the function. So not all the time you will have a matrix or a data frame. Most of the time you will have different kinds of data. And when you code for functions, you shouldn't be hard coding the format of your function. So a function that calculates Shannon diversity should be able to accept a vector, even if the input is a vector of abundances. Like if you put a simple input into the function, it should be able to return you diversity. So if the input of your function is a data frame, you can do one of two things. Either you embed into the function the possibility to make repeated calculations inside of the function. So the function itself, like, I will just write a little bit like Shannon. If you create a function here, you can have a parameter x that will be the input of the function. And this should be able to be a vector or a data frame. But at least a vector, at least one community, because it's the basic unit. So you don't need to make calculations element by element, but if it gets a vector, it's okay. And if you want to input a data frame, one of the options would be to use the family of apply. And I saw some of you using this. So for example, apply to the data frame along the columns, the function I wrote, the function. But also you could try to embed this apply inside the function so that if x is a data frame, it gets the calculation. So this is different ways of doing the things. But the key part is that at least for a vector, it should work. So because this will help you generalize the applications. If you, for example, if you put here a data frame that is 4 by 3, and inside your function you have a 4 and you have a 3, you're making it impossible to replicate, to be generalized, because you're hard coding some dimensions, for example. This is something that you don't want to do. So what you do is you create a function and you try to use it in different shapes of data and different dimensions. R does a lot of vectorization. I saw some of you were, for example, to make the binary table going element by element, and this is smart, but it's not needed because R actually works almost at the matrix level. Sometimes it will. You can ask if you multiply a number by a matrix, it will multiply everything. You don't need to go element by element. So so try to think about this. Maybe you will use these kind of things in the future or not. But I wanted to show you, yes, like some examples, quick examples of how you would create a function to create a Shannon. And this is the simplest way of creating this. Remember that we had Shannon was the sum of the relative abundances multiplied by some logarithm. It could be base 2, it could be base anything, but usually it's 2. So ln. And this is Shannon. So the first step is to calculate the relative proportions. So the relative abundance is simply, if you take a vector, you could just multiply the vector by the sum of the elements, and that's okay. It will give you a vector of relative abundances. You can check this in your code. And when we calculate Shannon, it's just minus the sum of this multiplied by the log. But when you calculate Simpson, it uses the same. It uses the same PI, the same relative proportion. But it does a different calculation. It's the square, the sum of the squares, and this is the diversity. So the next step in becoming better programmer would, for example, if you're already calculating PI for both functions, you could create a function that has the 2 indexes inside the same function. So instead of using only or my Shannon and my Simpson separate, you could have, for example, a parameter that is called index. And if this is Shannon, it will, like the function can calculate PI because it's the first step for both indexes. But if index is equal to Shannon, then you do one of the calculations, and if it's equal to Simpson, you do the other calculation. So this is more similar to what happens inside our packages. People do not create single functions if the framework is similar, okay? One thing that could be nice is you check when we do this kind of analysis. We use package vegan, and some of you used it already, and some of you already know its existence. So package vegan is made by some of the community ecologists, the most famous quantitative community ecologists in the world, including Pierre Legendre, who is like the author of Numerical Ecology. Take a look at the help and take a look at the code of the diversity function in vegan. Because some parts of it is going to be similar, but also when you code in R, you need to be able to foresee some errors. If the person comes with a character vector, the function has to return an error. So this is like the skeleton of something, but we are here in a space where we suppose that X is going to be a numerical vector. And inside of this room, it's okay. But in life, people can enter with character vectors or with things that are unexpected, and people who do the work have to foresee these kind of errors. So you will see that it has at the beginning some check that everything is numerical. If it's not numerical, some changes. So it's a good exercise to check how people do these things. So I won't go deeper in this part because I wanted most of all that to check how you think, but it's a nice exercise to check the help of the vegan diversity function. Vegan diversity. And the code in the repos. So, but last time I didn't even start with what biological diversity means. So today will be a little bit more talkative, and tomorrow we do a little bit more code. So basically, biodiversity is trying to account for the number of ways different elements in a set happen, and how frequent do different species, for example, in a sample happen. And it has two key elements every index has is the richness itself, the number of elements, of different elements that a set has, and the evenness, that is how even are the proportions when we calculate relative abundances. If we have two communities that have the same number of elements, for example, 5 species, the community that is more even, so we expect pi to be, for example, 0, 2, 0, 2, 0, 2, 0, 2. This is going to be more diverse than a community that has the same 5 species, but has an uneven distribution among them, because dominance here will be higher, and so it's dominance and evenness are opposed concepts. There are lots of people working on creating and analyzing diversity indices from the mathematical side of view from the mathematical side, because some indices were proposed in the past that don't follow some mathematical rules that a diversity index should have. For example, a diversity index should increase when elements of a new kind are added to a sample, but not necessarily. And of course, it will increase if it has more elements. The richness, if the sample has more species, the richness will augment. But this will change also the diversity, the evenness, naturally. So there's a lady that is called Sandrine Pavoine. She's a mathematical biologist. And among others, they have been working about how should indices be created. Because sometimes, and it happens a lot, for example, with Shannon, is that they are proportional to sample size. You have 2 samples, and you have a sample size. For example, I go to a forest, and the forest has lots of individuals. And I go to another forest, and it has less individuals. The individuals are more sparse. Without checking the numbers, I can assume that the sample that has more individuals will have higher values of channel diversity. And this is sometimes a mathematical artifact. You have to make them comparable. You won't be able to compare the diversity of uneven samples, at least not with Shannon diversity. Simpson is a little bit more robust about this because Simpson is equal to the chances that when you take two elements of the same sample, they belong or not to the same species. So we had this last time, and one thing that happens with taxonomical diversity indexes is that in 1973 there have been lots of, like in history, lots of indexes, and one of the proposers of one index was Regni. I don't even know how to pronounce it. Regni diversity, and Hill in 1973 unified the backgrounds, and he formalized it as some common diversity indexes are just special cases of Regni diversity. So if you look at the shape of the Regni diversity formula, and you think about A going from 0 to 1 to 2 to 3, you would find for n for 0 the Regni diversity and the exponent of Regni diversity would be equal to the number of species in the sample. And with A equal to 1, it's proportional to Shannon. And when it's 2, it's proportional to inverse Simpson. So we won't do this right now, but the idea is that it's a unified framework. It's a unified framework, and when you check and you calculate Regni diversities for a number of samples, for example, we have this, and I want you to do this. I want you to execute this on your computers. So please, you will have two communities, basically. And we're going to do this by hand. So we're going to do this, like we're going to create two communities, and the vector is the abundances of the species in this community. Okay? I can do this too. Check this. And if you haven't installed vegan, you can install vegan and you just check at the diversity function. No, they're not. They will. I will just open. Straightforward should have an H right here. I will come back. I will just come back right now. I will put this in my second screen. Here. Straightforward should have an H. Straight. Forward. Wait, let me check. I just have an H here. Okay. Wait, I will just, here. And I would like you to tell me the richness, of course. Sorry. It's still working. Sorry. So community A has 4 elements, and if you recognize or if you saw the vector that is resulting in community B, it has 8 elements, so it has a larger diversity. Look at the syntax of the repetition function. And I showed you here the bar plots that represent these 2 communities. So if you only, and this is something that people did in the past, if you only checked for richness, you would conclude that community B is more diverse because it has more species. But it definitely is less even. And when you calculate diversity in both of them, which is the values that you get? How much community A, Shannon, around? 1.11, 20? 17. And the second one? 1 point? So both is 1.17. Both of them are 1 point. I can't write here. Like 1.17, both of them? Okay. So if you used Shannon, it wouldn't even understand that they may be different. And when you check at Simpson, it's now you see the difference. And Simpson is a little bit more robust than Shannon for these kind of things. So and you see that there's a difference between the one that is like 1.90 something, 94, and 2.88. So most of the interesting questions in science do not stop here. Like, what is the most diverse community? Of course not. But this is part of the things that have been hunting people who work with communities in history. And I want you to execute and to check the help for the Renyi function in vegan, please. I will just open here my repo that should be the same as yours. And I will do this slowly. And if you want to, and if you are waiting, just take a look at the help. Not only the code of the diversity, but the help of the diversity function is just one of the best helps in the whole of R. I will close here. So library vegan. And what we want is something to compare the two Renyi diversities. So I have community A. I can't see anything. I should be. Okay, now I see. Wait. Community A is 10, 6, 4, and 1. And community B is 7. And the repetition of 1 7 times. And if I execute Renyi to community A, it will return to me a vector. Yes? It's a vector of values of diversity when we change A. Okay, so the default is here. The default is here in scales. And this is something that you can change. So it will return you, excuse me?
What are these scales?
It's the different values of A. It's the different values that of A in the formula for energy. This. Yes? So for 0, we expect this to be similar to the richness. For 1, we expect it to be the exponential of Shannon, and for 2, related to inverse Simpson. So this would be richness here, the first, and 1 would be Shannon, and 2 would be Simpson. But at the end, when we change A, what we are doing is we are going from a moment where every species has the same importance in the sample because it's richness. Every element of the species are giving the same importance. If you appear one time in the sample, you contribute to the richness at the end. And when we augment A, we are giving less and less importance to any individual species and giving more and more importance to the most abundant species. Yes, so nobody's going to use the whole, nobody's going to use A individually, but what people are going to use is the Renyi profiles as the whole profile, like how do they behave and how do you compare different communities at the end. So I have a vector for community A, okay? I will just save it as just let me, my only screen for this is this, so I have to write like this. So Renyi, community A. And the Renyi community 2, B, A, community B would be this. And how do we compare, how do we plot this at the end? And we can use one of the binding options. Because we have the same dimensions of both of the vectors. So we can do something like rbind, which is one of the functions. I will just put here the help. You combine the objects by row or column. So rbind of Renyi community A and R community B. It should work. Yes. So what I did was just binding them together. And this is Renyi of A and B, Renyi of A and B. And I want to plot this. If I plot this, it's not very beautiful because base R is not beautiful. It will just put maybe like some dots. Remember how does R plot diversity? And it will just do this. It will just put some points, and it's not nice. If there's a function that will allow you to plot a matrix, it's called matplot. But as you can see, if I do matplot right now with the current object, it is understanding each row as what it needs to plot. We don't want the eight indexes to be here. We want the 8 values to be in the x axis. So I will just transpose it with T. Or I could have done a C-bind to bind by columns. And it will plot the profiles. So it's not the best default because matplot is just made for matrices. So one of the options in matplot is to change the type. The type is P, and the P is going to be a character string. It's not nice. So I will just change it, and I will ask it to make some lines. Lines. Okay. What this plot tells us is that we cannot compare the diversity of these two communities because the two profiles of diversity, they intersect. So at the end, you cannot say which community is more diverse than the others. It's not as easy as to count species or richnesses or calculating one index. Are we there yet? Of course, you can change the labels and you could add some legend. When we're here, I will just check how you do this.
Andrea?
Hi.
Here. So, understood the scale parameter is like when it's slow, it gives more importance to the richness, to each individual. And when it's high, it's more about the most abundant, that's it?
Yes.
But it has other meaning besides this, or it's just a different way for you to weight these different things that you can weight when you're measuring diversity?
I didn't understand your question. Just let me. I didn't hear, but I didn't understand. Just tell me again.
So the scale parameter will change the way you calculate diversity, right? And when it's low, you give more importance to individuals and to richness.
Yeah, you give the same importance to every species, yes.
And when it's high, you give more importance to the most abundant ones.
Yes.
What I'm asking is if it has another meaning beside this, or if it's just a different way of giving different weights to different things when you're measuring the diversity index.
No, I think it's the only meaning. I don't know why. No, it's this. It's just a way to generalize common indexes.
Okay.
How big can this number be? Like, how big this number can be? Because we are saying that 8 goes here to 11. I don't know, but.
Oh yes,
64.
It's not 11. It's infinite. Oh yeah. So Matpot did something that is not nice. And it just, since the vector has 8 elements, it just calculated 8 elements. So this axis is not informative. It should be this axis, 0 to 64 and inf. Yes. So what we do here is that we can tell it to take the axis off. Axis equal false. Should take them away, and we want a correct axis. So I will create a box around it, just to have the box itself, but it's optional. And the axis, you can paint it manually. Unfortunately, because it's matplot, because it's just a dump function that will just put the index of the version. I am not doing this in ggplot, but you would have to also tell it that A varies from 0 to infinite. Of course, yeah, I'm fine. Is it large enough? Oh yeah, sure, you can do this, of course. So I just took out the axis, and the first would be the margin. It works the same. 1 would be the x this side. 1 would be the x, and 2 would be the epsilon side. So I will just add the axis for the y without thinking much, because this is the true values. And if I want the axis for the y, I just execute axis, and I say like the side should be 1, and the labels here are the important ones, because the labels are these elements. So it should be 0, 0, 0.5, 0.25. Let me certainly 5, 0, 5, 1, 2, 4, 8, and infinite. If I put infinite here in text, what will happen is that the whole vector is going to be a text vector. Look at it. I cannot mix types in R. But that's okay. I could have put here the quotes, but since I know that R will transform it into character, I just put the quotes in the last one. But I know it's not going to be a character, a numeric vector. And the last, if I do this, it will say, hey, you didn't tell me where to put it. You didn't have anything at. So one of the parameters of this function is at. And at is basically 1 to 8, because it's going to be even. Oh, no. Oi.
I think it goes 1, 2, 4, 8, 16, 32, until 64.
I can't hear you. Ah, 64, of course. I'm missing lots of labels. Thank you. I was just like, why did this happen? But this happened because it's 16 and 32 and 64, 3 and 64. And inf, and now it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So it's 11. So the 11 part that Sebastian was asking actually is just the index. Oh, you don't want to do this. I will just replot it. Okay, so this is it. So A can vary until to large numbers. And of course, another way to plot it would be to respect the numerical side of this, but this is not today. So this is just a way to tell you that taxonomy diversity indexes can be very wide. I will put a series of references for those who are interested, but at the end, it's largely, for some people, it's largely a math exercise, because you know how a diversity index should work, how it should be monotonic to some of the properties of the communities, and how it should only vary with the dimensions that it's supposed to vary. So when people propose new diversity indexes, they have to be very careful with this formulation of. Are we okay? I will just to check what I am doing here also is like checking, showing you how plots also work here. So we can add a legend. We can add a legend just by using the legend function. And basically what the legend function works is you tell them where you want the legend. It's X and Y, but also it can accept things. It accepts character vectors, just as I can say, please put it in the top right corner. Right corner. It wants to have a legend, actually. So the legend would be legend. A vector of the numbers of the groups you want. So community A and community B. And the line type, because I asked it to plot as a line. the default line types for R, it's not here, it's in plots here. This is what line type 1 means in R, and this is what line type equal to means in R. So it's continuous and dashed, one kind of dashed. So line type 1 and 2. And the colors also. The first color is equal to black, and the second color is equal to red. And this should be an equal. I will just format this nicely. Okay, so this should add a legend to this plot. And as you can see, the plots in base R just can add some elements, just like ggplot works. The same as ggplot works. So, but just let me know if you have any questions, if I can go on, please.
My labels are not changing.
Your labels are not changing. Did you execute again?
Yes.
Did you? Is it superimposing? Is it writing again on top of it? So one of the things that happens when you plot things in R is that some functions call for new plots. So for example, if I delete this plot, if I just close this device, I will just close it now with def off. If I execute box, it will tell me error in axis. Plot new has not been called yet because axis is not one of the functions that calls for a new plot. And box is neither. Box also does not know where to plot. They only work when there's already a plot inside it. Legend is the same. Whenever you see plot new has not been called yet. So maybe what you're having is that you're plotting on top of it without changing. So try to come back to matplot. Matplot is one of the functions that create new plots. And check if the labels at that point are changing. Are we? I want to change the label on the y-axis, for example, too. So here, y-lab, range, diversity. Did it solve to replot? It didn't? So just let me check. What's this force? How are you? Labels at 1 to 11. Just execute it again. But are you having? Just let me check here. Why? This? Because the side is the other one. So this is your side 2, and this is your side 1. And I think that since you had like 1 to 11, and this is like 0 to 1, it wasn't showing. Okay.
Why they're superimposed?
Because it superimposed because I replot it instead of plotting again. If you do this again, with changing, it will just add to the same plot. So it was a question of margins. The side 2 is the epsilon, and the 1 is the x.
Andrea, I have a question. I'm not familiar with these kind of studies. So what kind of organisms I use? Because I need to have like a threshold of the size of the organism. Otherwise, I'm going to include, if I include very small I don't know if we include, for example, microorganisms or very small things. Is it a good thing to do, or I don't know, what do you do in the field? Or can I compare different communities composed of organisms of different scales? Should I reconsider my things inside the functions when the organism is very small, so I can expect very big numbers? I don't know.
Okay, I think you're pointing to a problem of definition from the starting point of community ecology. So the basis here is community ecology, and community, the definition itself of community is a very tricky one, because at the end it's an operational definition. You work with the community that you are interested in, and so community means in biology populations of different species that coexist together in the same site. So first it has a geographical frontier that is tricky, because you cannot measure everything everywhere. So it's operational in terms of what do you mean by site. They coexist where? So you can have different scales, spatial scales, and spatial scale in ecology is just like a large topic. But also, what is a community? Depends on the kind of questions you have. So you can have several species of the same trophic level coexisting together, and you want to understand, for example, the relationships of competition or facilitation between them. So you only have plants, or you only have insects, or you only have large trees, because the definition and your question is related to the large trees in the site. So, for example, if you are working with microorganisms, you can define that you will sample a pond with some sampling techniques that should give you significant samples of the organisms in the place, and depending on the depth, some variables that change when you do this kind of sample. And you can. Excuse me? It's arbitrary it depends on the scale and the question that you're asking. So communities, one of the questions that I don't know, and people, is like, do communities exist in nature? Do populations exist in nature? And it's like, yes, but do communities? Yes, they do, but of course you know that there are some different coexisting organisms. But when you work with them, you cannot define community as everything that exists together, everything in the same place. So it changes a lot, and it's one of the key problems that we have when sampling things. So, for example, with trees, I can have a large amount of forest, and the trick is to do sampling designs that give me a good glimpse at the diversity of organisms in that forest, and we need to use. For example, when it's trees, it's the minimal size of a tree. And you say, I am going here, I'm going to sample everything that is 2 centimeters dBH, and the rest I am going to lose it. When people work with animals, they know that the detectability of the animals is very different, so their questions change a lot. The sampling device can or not attract or having a mesh, you have a diameter that is going to be lost. So it's before. It's before. Yeah. Please.
The way that we add the numbers in the vectors for community A and B must be in a decreasing way, or I mean, because in community A we add 10, 6, 4, 1. Or
No, not at all.
It doesn't matter the order.
No, it doesn't at all.
But it doesn't alter the result of the index?
It doesn't, because it's only taking the proportions. You can do the same exercise with the community.
Okay.
It will just go via the formula, and the relative abundances won't change. So this is an example with two simple vectors. But you can do this with the COM matrix that has 57. You don't need to reorder anything. You just execute Renyi for all the community. Okay. This was just an artifact of how you built it. So when we talk about, I will just come back to the slides here.
Andrea?
Of course. Okay.
Because I'm having a bit of difficulty to understand what is happening in the plot. Could you repeat about the two lines and the interaction between them?
Okay. In this, at the end, the Renyi, at the end, what we have here is different levels of Renyi diversity. That is not exactly the number of species, but for example, for 0. It's related to the species richness. It's because it's the exponent. Let me check. So we have diversity when everything is taken into account. So like richness, and we have one. It's Shannon, which is very used, and when we have two, it's related to Simpson. Yes, I think the fact that it's Renyi versus Hill numbers, that there's an exponent, is the way Renyi calculates. So it's in the same scale. I will just do this. Where's Renyi? So Hill falls. So this is what happens. Like, we are not calculating the Hill numbers. We're just the Hill numbers would be richness and diversity in different levels, but and Hill numbers are, remember, the Hill numbers are the exponent of this. So this is why this plot is still in the original scale of diversity, which varies from. It does not vary, it does not have an upper bound. But it's not in terms of number of individuals. If we made this with Hill equal true, let me see. When is Renyi? Here. I will just duplicate this. I will create Hill. Look what it does when I create for Hill numbers. The first element in the vector is the number of species. Yes, and the element for A or alpha equal 1. It's the exposure. It's related to Shannon, but as in it's the exposure of Shannon. Okay, so at the end, the number of Hill, there's something extra in diversity theory that is called equivalent numbers. At the end, when you want to compare some different communities, you want to know how comparable they are. There's something that is called like the equivalent numbers. So in this sample, even if it's 20 species, I have the equivalent of 17 species functionally. For example, this is done with functional diversity. So it's different ways of calculating the taxonomic diversity, giving more or less importance to each species. Okay. So one of the frameworks that unified taxonomic diversity was the Hill numbers and Renyi diversity. But when we talk about functional diversity, can I just Is it fine? Are you okay? Are you not okay? It's just because I know that I told you earlier to go to the help of diversity function. And one of the problems that we have in this area is that people use these indexes that they like. So for example, the help itself says the following thing. Better stories can be told about Simpson's index than about Shannon's index, and still grander narratives about rarefaction. However, these indices are all very closely related, and there is no reason to despise one more than the others. But if you are a graduate student, don't drag me in, obey your professor's orders. So it's a little bit depending on the school, and it's a little bit depending with who you're talking to. But at the end, what I wanted to show you is that there's a unifying mathematical framework for all the diversity. It's related to information theory when you talk about Shannon. It has mathematical formulas close to entropy, and we are going to talk about another unifying framework that is Rao's quadratic entropy, that is similar to this, but not necessarily. And part of the sensitivity is that some indices are more sensitive to rare species or to abundance. So this is why we calculate the whole array of indexes. When we talk about functional diversity, I don't know if any one of the biologists here has worked with trait-based approaches with functional diversity here. I think maybe Thales has. Like functional diversity, are you familiar with?
Functional traits, but not with functional diversity.
Okay, so functional traits are what we measure when we calculate functional diversity among others. And the whole thing about functional approaches and trait-based approaches is that until now we have been using the species as an analytic unit that is valid in itself. Species exist, and one species is different from the other. And what happened is when we talk about diversity, if you go back to the first definition of diversity that I put here, diversity is complex, and it also is related to evolutionary units and to traits. So we know that even if two cactus species are two different species, functionally they are super similar. So a community made of only cacti species is less functionally diverse than a community that has different life forms or different heights, different. So when we were measuring diversity, lots and lots of calls were made of, we have to go beyond taxonomic diversity. We have to go through the phenotypical characteristics that allow species to belong to the environment, to thrive in it, to replicate in it. So what we measure when we measure functional diversity are functional traits of the species. And this is the briefest introduction to trait-based approaches in ecology you will have. It's like 2 slides. Because basically a functional trait is a morpho-physiological or demographic trait of a species that allows it to belong to the environment, to respond to the environment. And it will determine the species' ability to survive. So species are not there because of their belonging to a species or to the other. It's because they have traits that allow them to survive or not. And we have some traits that are correlated with others, and there are some fundamental trade-offs that make it impossible for a species to invest energy in all of its parts at the same time. You could ask, why does not a species create lots of large seeds and frequently? It's like because it's energetically impossible. You have to choose, and that's why ecological strategies exist. So some of the traits, individual traits, are correlated with the others because of these trade-offs. So in general, species that produce a large quantity of seeds, for example, they do not produce large seeds. Large seeds come in smaller amounts, and that happens with animals too. This is a simplification, but you cannot put a lot of energy into both doing large propagules that required a lot of energy and make a lot of energy. And if you are in an environment that is harsh, that requires you to invest energy in survival, even less. You won't be having an investment in energy for doing either frequent or large seeds, for example. And functional diversity is at the end in a community that is defined arbitrarily depending on your question. What is the space that is occupied by the different traits of the species? You have and it's related to a function. So you will have a number of different functions, different types of functions, and different distributions between the species. Trait should be measured at the individual scale, and this is key because we can have inside the species, we can have intra-specific variation. Not the traits are not like one, conserved at the species level necessarily. Sebastian?
I have a question with the organisms that are, like you don't have a single organism but a colony. For example, ants. If I'm measuring, I don't know, I'm going to say that my community is composed of insects and plants, I don't know. But I'm going to count ants. Is it worth to count individuals if they are always in colonies? Should I count the hormigueros or only the individual? Because it doesn't make sense for me to count an ant because they are a colony of organisms. How can I deal with this? Maybe the people who work with this, they know, but
I don't know if somebody works with insects
or
here.
colonies of things.
We have clonal plants, for example. I think at the end it's the formulation of our question, because if you are trying to study different ants in communities that have different species of ants, there are some questions that make sense to count, like density of individuals per species, for example, but some of the questions won't even need this differentiation. For clonal plants, for example, you can understand the individual as a sole individual that has different manifestations, like two clones of the same plant can be competing between each other or interacting with others. But they may share traits. For example, it's okay, you can measure it at the individual scale, but at the end it's okay if the whole population has the same trait. It just is not a good pick for your question if you have a trait that does not vary among your individuals. So it depends, it's a little bit of a match between the question and what you pick as a functional trait. If I want to differentiate the different, for example, ants in a colony, what are the possible ways to differentiate them? They all will have the same morphology. Maybe their roles in the colony, the ants and the soldiers, and maybe their behavior would be more interesting to analyze than morphological traits itself, like fast and like behavior, like how they gather or not. So it depends on what you choose and what you ask. But yes, we deal with these kind of things. Like this is not even an individual we have sometimes. This is the same individual. You go and it's like a mat of plants that is the same, it's a clone. And they might be expressing parts of the phenotype differently, even if they're clones. But at that point, you have to decide how are you going to treat them. Sometimes you treat them as different, sometimes you treat them as the same thing. So it's very tricky in terms of the definition of the problem. Like volvox and things, yeah. And when we measure functional traits, they can be contiguous, like the height of the individual, and they can be categorical, like the preferring the alimentary preference, if an animal is omnivore or carnivore. It does not have a value in itself or an order. It's just different categories. So when you have this kind of mixed data, you need to have correct like distances and dissimilarity measures that take into account these different ways of measuring traits. So one of the most used dissimilarity indices is the Gower distance that can lead with binary data, ordinal data, categorical and ordered, continuous data, and with binary, I already said. Okay, and it will deal correctly with different scales of the variables, because you cannot compare height in centimeters with number of seeds and assume that the numbers are the same. You just have to scale things. Some of the examples of the traits can be whole individual trait. I have a definite bias toward plants, sorry. So whole plant trait, but whole individual trait, such as the height, the growth form, their behavior. Or you can have parts of the individual be measured. So in the case of plants, for example, leaf traits are very key to determine the economic of the individual, the economy. And since plants are modular and plants can allocate in time, they can change their allocation. The leaf trait is very useful because the same plant, same species, same genetics can respond differently and have different traits in different environments, for example. One of the earliest functional diversity is Village from 2008, and it's very geometrical. What it does at the end is that it plots in two-dimensional or tri-dimensional spaces the n-dimensional space that are occupied in traits by the individuals of a community or of a species. So what we have at the end is, we will see tomorrow the traits matrix of the CESTAS dataset that you have. At the end you have, for example, you have 13 traits. When we talk about multi-dimensional spaces, it's just like 13 axes, potential axes, in which each individual is located. So most of the problems in functional diversity, when they are led like this, are dimension reduction problems. You want to reduce the 13 traits, the 13 dimensions of variation, into a way of graphically and mathematically lead with this in less dimensions. So this example is only with 2 traits. So you have trait 1 in the X and trait 2 in the Y dimension. And you have 9 species located in the functional trait space of created by trait 1 and 2. This could be species, but also this could be individuals for the same or different species. So in this case, it's species. The trait is at the species level. It does not take into account intraspecific variation. And what we call functional richness is the whole area created by a convex hull in this n-dimensional space. So for example, in this data set, in this example data set, the richness, the functional richness is 1.5. And another community can have a different convex hull, and a different functional richness at the end. So richness, instead of species richness, which is different numbers of species. The fact we have lots of traits and we have lots of individuals. So we cannot say like number of traits because we haven't measured everything. Just in the space that we measured, functional richness is the size of that space, the size of that convex hull. And functional evenness, what the authors did is they create a minimum spanning tree between the species, and they say this is the shorter path that connects one-to-one all the species in this bidimensional space. And this is a way, this second one, okay. This second one is a linearized Minion spanning tree. It's the same figure, but this was in the trait space, and this is not in the trait space. And the evenness is how the length of these segments vary between each other. If you have a species that is very different to the others in that trait space, it will position itself very far away, and the segments will not be even. You will have a large tail, you will have a large segment that goes away and connects these species. So how even are the segments that are connecting the species in the trait space is one of the measures of evenness. I'm saying one because there are like lots of authors working on this. And the third element of functional divergence in this framework is functional divergence. So how far are they from a mean trait space? So this is like a mean, and how far are you away from this is part of this deviation is part of how diverse is your community. This functional richness, evenness, and divergence can be calculated in package FD. I haven't used it because when I started working with this, I wanted something that could be unifier, and this is a very different framework. It was super used to analyze functional diversity, but since it's a mostly graphic method, it depends a lot on which traits do you measure, how do you measure them, and it has no way to go back to taxonomic diversity in a way that is a similar framework. But this is calculated in FD package, in R, functional diversity. And when we talk about phylogenetic diversity, we are talking about the fact that species that share common ancestry are like all species come from a shared common ancestry, and in any community you will have species that are closer phylogenetically from the others. So in diversity theory, if you have a community that is composed by very closely related taxa, it's less diverse than when you have a community that has elements that come from different parts of the phylogenetic tree. The most simplistic way of seeing this is counting the number of nodes that separates any two species in the community, which is just for the simplification. So if you have a phylogenetic tree and you have some nodes separating the species, like 1, 2, 3, 4, 5, you would say that these two first species are apart from one node, and that these two species are apart like one. It's weird because this changes, but like you could say like, yeah, it's further than this. And you have pairwise distances. So the number of nodes is not ideal. The ideal is that you have a time, that you have a calibrated phylogeny that you don't always have, and that you have a notion of the distance in time of the most recent common ancestor between any species. A first classic measure of phylogenetic diversity was a dendrogram-based approach. It just took the length of the branches and said like, and just sum them. It was the sum of the length of the branches in a dendrogram. So if you have two dendrograms, the phylogenetic diversity of the community that has the red elements would be higher than the than the phylogenetic diversity in the blue clades, because you don't sum this large branch a couple of times. As usual, as with most diversity indexes that are created from scratch, it is an index that does not work numerically and does not work to unify with taxonomic diversity. And it depends a lot on the way you create your trees. So it's not exactly a way to summarize phylogenetic diversity. But what Faith was doing in his paper was giving importance to isolated clades in communities. So for example, if you have this kind of long branches that are isolated, if you have an individual from a species that has no common relationship with the others, or that has a very distant early divergence, the presence of this kind of individuals in communities should be valued more than only richness, for example. So it was a way to give importance to the distinctiveness, the phylogenetic distinctiveness on top of the diversity, not only the richness. In community ecology, I don't know if we have bioinformaticians here, I think. Sometimes when people work with genomic and genetic data, they create their own, their work is creating the phylogenies, and taxonomists do create their own phylogenies. But when we are in community ecology, most of the time we don't create them. We don't have even the funds to do the molecular part to create a phylogeny that is related to what we are seeing on the field. In the ideal case, you would go to the forest, and some labs do this. Like, you take samples of everything that you see that you want to study. You do the molecular part and you create your phylogeny. But this is not a reality in lots of works. And so we have topologies of trees, of phylogenies created by others, or like standardized. So for example, the fact that the APG tree exists for flora, for trees, and you have some dated nodes, you basically can have estimates of phylogenetic pairwise distance between any two individuals in a community. And there are some algorithms that help you with calculating the nodes of the dates of the nodes that are not dated. So sometimes you just split. If you have here a dated node and you have here a dated node, any node here will just, for example, be split in equal sections. So you know, so you don't know exactly what the age of the node is, but you have a way to compare different nodes. And one unifying paradigm of the diversity, taxonomic, functional, and phylogenetic, at the end we're talking about distances, pairwise distances between individuals of a community. So a unifying paradigm that has been proposed is one of the early diversity measures, that is the Rouse quadratic entropy. And if you see, it looks a little bit, you're summing relative proportions, but it has a pairwise distance between individuals of the community. So you sum for every pair of species, you multiply their relative proportion and the distance, the pairwise distance between them. And the trick is that in taxonomy, the species, the pairwise distance between any species is a matrix that only has 0 and 1. So you have, if you have two species that belong to the same, two individuals that belong to the same species, their distance is 0 because they belong to the same species. If you have 20 individuals that belong to different species, the distance between them is 1. So this is a pairwise taxonomic distance for the communities. But this, the fact that you have pairwise distance between individuals, allows you to use other kinds of distance for functional, for example, Gower distance for functional traits for every individual, and phylogenetic distance when you want to start studying phylogenetic diversity. So Rao has been more of a unifying paradigm to compare what taxonomic, functional, and phylogenetic diversity would mean in a community. And at the end, it works when you have this kind of matrix in taxonomy. It's equivalent to Simpson's diversity. It just gives you Simpson's diversity at the end. It's just, since it's multiplying PI, it's giving you the chance that you take any two individuals from the same sample and they belong or not to the same species. This is how Simpson diversity works. It's just like, if I take right now two individuals from the same community, are they or not? And they give you, and Simpson gives you an expected distance of the individuals. At the end, it's a probability that they belong to the same species or not. The same holds for Rao in the distance, pairwise distance. When you use Gower's distance in the trait space, or you use phylogenetic distance in the phylogenetic space, you will have an expected difference of any two individuals in the same sample from the set. And that you can start comparing between taxonomy, functional, and phylogenetic diversities. So we're going to talk a little bit more about distances tomorrow, but this is basically the quickest, briefest way, and it's not brief, way to talk about these facets of diversity when we analyze it. So I know this is too much, and I talked a lot, but in community ecology, we're going to talk a little bit more about distance measures, and so far it has been pairwise, individual and species-based, pairwise distances, and other approaches in community ecology are related to the pairwise distances between sites, for example. And we will have the scale. You could have pairwise distance between individuals of the same species, or between individuals of different species, or between sites. If we will have a lot of distance measures to analyze the differences between individuals, species, and sites in this whole framework. And I will stop now. But I want, no, like, tomorrow we are going to do more code-based things.
Geç Kumic.
Excuse me?